So Mary was naughty and had a child with Bob or Larry. But was it Bob or Larry? Now, if you're on the website, you can see the diagram with Mary, Bob, Larry, and the child and their DNA analysis. And it's on the genetic fingerprinting page. You can search for it on the website. And we're going to talk about how you can use genetic fingerprinting and analysis of DNA to establish blood relations between people. And for Mary to solve her conundrum. Despite all the vile racism and sexism in the world, sequence-wise, DNA between humans is pretty much identical. So how can you tell the difference between blood relations? Well, 0.01% of the DNA is different, and that is enough. A lot of the human DNA is just repeating and repeating sequences that don't code for protein. And it's those sequences that are used to analyze the relationship between people genetically. So you take a sample from a person, you multiply by the polymerase chain reaction, that is a separate topic, I think, already on the website. And why do you multiply it? Think. You have to multiply the sample, because if you don't, it will be so tiny that whoever's doing this analysis in a lab is going to put it in a tiny little tube. It's going to stick to the tube, and then the idiot is going to drop it on the floor and lose the sample. And how are you going to get your grandma to spit in a stupid thing again? After you've got a load of this DNA, you put a load of enzymes in it that chop it at specific sequences to create a multitude of different small little fragments. And then you tag the fragments with a fluorescent agent that is then visible as used on a photographic film, and you get bands. And those bands are those fragments that you just chewed up with the enzyme of the DNA. Now, these fragments will be different sizes depending on the sequence, because the enzymes that chew them up are sequence specific. So if you have the same sequence as your sister, then the bands will be the same. It will be cut at the same places because the sequence is the same. If you and a stranger off the street compare it, you might have a different sequence. So where one enzyme cuts your DNA at that point, it might not cut their DNA. And where it doesn't cut your DNA at another point, it might cut theirs. So you would get bands at different positions, different heights on that film. Now, when doing a paternity test, obviously not all of the DNA of the child is going to be the father's. So you have to account that half of it is probably going to match, well, should definitely match the mother, and the other half is then to be compared to the candidate fathers. Jeremy Kyle, come in, my friend. In this particular diagram, if you just so happen to be on the website, we have the lines lined up, and the first column is Mary, the mother. The second column is Bob, potential father. The third column is Larry, potential father. And the fourth column is the child. And the child has got 4 bands, and one of them match the mother. So the other 3 are compared to Bob and Larry, and Bob has got 1 line in common with the child, and Larry has got 3 lines. So Larry has got 3 out of 3 lines the same, and Bob has only got 1 out of 3 the same. Obviously, Larry's the father. Well, unless he's just an incestuous grandfather, of course.